but methylated cytosine is not converted to uracil. So since methylated cytosine is not converted to uracil, that change is detected by MSP. MSP is methylation specific PCR. So it was performed after extracting DNA from treated and untreated harvested cells, and then time course study was done. It was done by treating cells with a fixed concentration of compounds over different time periods, like we took 48 hours, then we took samples for 72 hours, and then finally for 6 days. Finally, we studied the reactivation of methylated tumor suppressor genes. In case of any reversal of hypermethylation, semi-quantitative reverse transcription PCR was used to check the reactivation of the methylated tumor suppressor genes. Now, how we did this? First of all, we carried out the MTT assay. You can see MTT assay was to estimate cell viability after the treatment of CH cells. You can see these pictures or graphs of CH cells, and this is representing the IC50 value. This is after treatment with curcumin. The IC50 value was calculated based upon this. This is for genistein, capsaicin, EGCG, which is present in green tea, resveratrol, azadirachta indica, as I already told you. This is neem, Osimum sanctum. This is tulsi. In India, it is called as tulsi, and VID20181022110313. somnifera, this is called as ashwagandha in India. Similar studies were carried out for HeLa cells also. You can see these graphs helped us in calculating the IC50 value. That is the concentration at which 50% of the cells get inhibited. Now these are the IC50 values of different natural compounds and plant extracts in CH cell line. You can see for curcumin it was 50 plus minus 2.51 micromole. Now what is this? Well, what is this? Is that, see, I would like to tell you, 50 plus minus 2.5 micromolar. This is because IC50 value is calculated to be 50 plus minus, this is the standard deviation. So we repeated these experiments for 3 times. Thrice we did all these experiments, and we calculated the standard deviation also, so that there are no chances of error. So we did this for all the compounds as well as the plant extracts. You can see this. And then we went for the morphological changes in CH cells after 24 hours of treatment with the test compounds and plant extract. See, you can see this clearly. This was the control in which no treatment was given. And you can see this. After treatment with curcumin, apoptotic bodies are formed. You can see this. These are the apoptotic bodies. After treatment with genistein also, apoptotic body formation is there. Then allyl sulfide, it did not show the formation of apoptotic body because it was not found to be, that toxic for the cancer cells. Capsaicin also resulted in the formation of apoptotic bodies. EGCG, you can see this one. These are the apoptotic bodies. Resveratrol also. Then azadirachta indica, Osimum sanctum, and VID20181022110313, somnifera, they did not result in that much amount of formation of apoptotic bodies. So that was the thing. And similarly, these morphological changes were studied in HeLa cells also after 24 hours of treatment. You can see this is the control in which no treatment was given. And here you can see after 24 hours of treatment with curcumin, these are the apoptotic bodies. Yes, you can see this. A huge number of apoptotic body formation is there. Then after treatment with genistein also, these are the apoptotic bodies, you can see. And allyl sulfides, it is exactly similar to the control. No change after treatment with allyl sulfide. But here after treatment with capsaicin also, some apoptotic bodies were observed. After EGCG, you can see this. Then resveratrol also showed the formation of apoptotic bodies. Some of the apoptotic bodies were observed after treatment with azadirachta indica, Osimum sanctum, and VID20181022110313 somnifera also. Now coming to the DNA fragmentation assay, as I told you, DNA fragmentation is called as the hallmark of apoptosis. So you can see the induction of apoptosis in CH cells after 48 hours of treatment. First lane number 1, this one is representing in the first gel picture. The first lane is representing the control. The second one is after treatment with genistein. You can see the DNA is fragmented. And then third one is after treatment with allyl sulfide. And fourth one is after 50 micromolar treatment with curcumin for 48 hours. You can see the formation of DNA fragments. Similarly, you can see the formation of DNA fragments after treatment with EGCG. That is the fourth lane. Then fifth lane is for azadirachta indica, as well as sixth one for resveratrol also. The treatment was given at IC50 value, which was calculated based upon the MTT assay. Now, the induction of apoptosis in HeLa cells after 48 hours of treatment, you can see this. The first lane in the picture number, this picture is having 3 parts, A, B, and C. In the A gel, which is represented as A, you can see lane number 1 is representing the control, lane number 2 is representing genistein, and you can see the formation of DNA fragments here after treatment with 45 micromolar of genistein, and here you can see allyl sulfide, it did not result in the formation of DNA fragments. This correlated with our study in which we could not find the morphological changes, formation of apoptotic bodies after treatment with allyl sulfide in ZIA cells also. So similarly, this was the case in HeLa cells also. Allyl sulfide did not result in the formation of any DNA fragments. But yes, coming to the fourth lane, you can see curcumin resulted in the formation of DNA fragments. Similarly, you can see in this one, B, lane number 1 was after the treatment with resveratrol, showing DNA fragments. Then lane number 3 after treatment with EGCG is showing DNA fragments. And amongst the plant extracts also, in C, you can see Osimum sanctum is the first one. But the third lane, that is Azadirachta directa indica, and this one, Methania somnifera, they resulted in quite a bit formation of DNA fragments. Now coming to MSP. After DNA fragmentation, we went for bisulfite modification and we performed MSP, that is methylation specific PCR. Now this is the basis of methylation specific PCR. This is a technique which can